continue with lecture 3, I'm going to finish up by talking about the internal structures of bacteria. So the cytoplasm is basically where all the internal structures exist. It's sort of the jello-like solution. It's mainly made up of water, but this is the site where a lot of biochemical processes happen, where the ribosomes are located, where the nucleic acids are located, and any other internal structures that I'm going to talk about. So bacteria have single circular strands of DNA as their bacterial chromosome. Some of them have more than one, but it's most typical that you have a single circular strand. And there's no nucleus in the bacteria, so the bacterial chromosome tends to sort of congregate and bunch together in one area of the cell, and when it does that, it's called the nucleoid. So here in this picture, you can see that it's stained, so where it's glowing bright yellow. That's where the genetic information is, or the nucleic acid is. So in addition to the bacterial DNA, there can also be non-essential pieces of DNA. So they're not essential for the life, but they might have some piece of genetic information that makes it beneficial for them to survive in certain environments. So it's typically like a round, circular, small, double-stranded piece of DNA, and that's called the a plasmid. So here you can sort of see them here, and they're not necessarily drawn to scale here. This is a lot larger than these actually are. A lot of times, some things that they can carry that are like non-essential are like antibiotic resistance or the productions of toxins or enzymes or things like that. So small genes that make proteins or do something beneficial for the bacteria, but are not necessarily needed for it to survive under optimal conditions. Now, this plasmid can be transported from one bacteria to another through the process of conjugation using the pili that we talked about previously in the lecture. And not all bacteria can do this, but some can communicate their plasmid from one bacteria to another, and this is how antibiotic resistance can then spread within a population. If you look down here, you can see that these are actually plasmid maps. Plasmids are also used when we do genetic engineering, and so you can purchase plasmids from a company, and they contain different sites, and you can insert a gene into these sites and use them for inserting a certain gene into an organism that you want. So for example, say we wanted this bacteria to make insulin, I'd buy this little plasmid, I'd cut it, and I'd insert the human insulin gene, and then I would make this bacteria take up that plasmid, and then that bacteria would then be able to make insulin. Ribosomes are found in the bacterial cytoplasm, and they are a little bit different than eukaryotic ribosomes. Again, we try to identify things that are different about eukaryotes and prokaryotes because it is potential drug target. So a ribosome is made up of RNA and protein. The RNA is called ribosomal RNA. It's a structural RNA. And the ribosome of a prokaryote is a 70S ribosome, as opposed to eukaryotes are 80S. And if you look here, if we divide it into its two parts, you have a large subunit that's 50S and a small subunit that's 30S. So when I took my biology classes, I was always taught this, but nobody ever told me what that S meant, what that S unit was, and I was always confused by the fact that together it was 70, but separate it was 50 and 30, which adds to 80, but it's not eukaryotic, which is 80. So essentially what the S unit is, it's a Svelbarg sedimentation unit, and essentially if you were to put these ribosomes into a liquid and you were to centrifuge them, it's how easily it moves through that liquid. So it has something to do with its shape and its ability to pass through a liquid, which is why they can have these seemingly nonsensical S units that don't always equate to one another. But just keep that in mind. Prokaryotes have 70S and eukaryotes have 80S. Some bacteria can have inclusion bodies, and an inclusion body is essentially a little packet where nutrients are stored. So if you're going through a period where you have a lot of nutrients available to you as a single-celled organism, in order to ensure your survival, sometimes you'll take that excess nutrient and you'll store it so that it's available in times when nutrients then become depleted. Um, some interesting adaptations is some aquatic microbes can actually store gas, and so it helps them to control their buoyancy in the water so they can move up and down in the water column, changing what temperature they're experiencing, what availability of oxygen they're experiencing, availability of sunlight. So it's kind of a neat adaptation. Now, a specific type of inclusion body is a granule. A granule is given its name because it stores inorganic compounds, as opposed to inclusion bodies just in general typically store organic compounds, but granules specifically inorganic compounds. So some things they like to store, for example, like iron oxide. Iron's a really important coenzyme for a lot of metabolic processes. So storing of iron is important for a lot of bacteria. You can stain granules, so you can usually see them in microscopy. We call them metachromatic because they'll stain a darker color when they're in the presence of methylene blue, as an example. This is actually being stained with some fluorescent sort of stain. I mentioned bacterial motion when I talked about flagella, and I said that some bacteria can be magnetotactic, which means they move towards or away from a magnetic field. And the way they do this is by storing iron, which is a magnetic metal. Now, in order to establish the structure of the cell, some bacteria contain actin cytoskeleton. So actin is just a big long polymer, and here you can see just the actin being stained within the cell, and all it does is really contribute to cell structure. It's just like scaffolding. Bacterial spores, or endospores as they're called when they're inside of bacteria, are only formed by certain species of bacteria. Bacillus species in particular are the ones that we'll look at in the lab, but also Clostridium and Spore sarcina species. So when nutrients start to deplete, basically it's a defense mechanism that a spore is formed. And the spore essentially stores all the genetic information, and it's like a seed for the bacteria that can then hatch later and form a vegetative cell. So when it's inside, it starts to form inside when nutrients are depleted, and you call it an endospore. Then the outer vegetative cell will die, and then it will be released out into the environment. When it's released out into the environment, it's then called a spore. And once the nutrients then become available, that spore essentially hatches or germinates, and it then forms a vegetative cell or a new living cell. Now, endospores, we talked about the difficulty of killing them. We talked about Louis Pasteur and Tyndall and Tyndallization process. They're one of the hardiest life forms, and they can withstand huge extremes in temperature and radiation, chemicals. They're extremely heat resistant. This is actually a picture of scientists taking ice cores in the Arctic and the Antarctic. They do these really deep down ice cores, and they're able to discern something about the environment when that ice was frozen in that area. So the deeper you go, the older it is, and we've been able to find endospores that are over 250 million years old that we've then been able to hatch in a lab. So kind of crazy. This is what they look like under the microscope. We'll be using colors, and this is just black and blue. The colors you'll see when we do it in the lab will be like red and green. But you can see here is an endospore. It's still sort of contained within the bacteria, but then once it's released, so these are spores. And when we stain in the lab, the vegetative cells will be one color, and the spore or endospore will be a different color. So you then can germinate them, and that happens in times when now water is available, or the correct temperature is available, or there's nutrients available, and they can germinate very quickly. And there is a huge medical significance to spores, because spores are what can cause a lot of infectious diseases. So, like Bacillus anthracis, so anthrax. The anthrax attacks that happened after 9/11, they were anthrax spores that were then inhaled, which caused people to get sick and some people to die. Other diseases like tetanus and botulism and Clostridium perfringens we talk about, I think, if I remember correctly, causes gas gangrene. So it's the spores that are the causative agent that makes the person sick. This is tetanus, and this is botulism. So the last thing I'm going to talk about is bacterial shape and arrangement, and I'll do that in a really quick wrap-up video.